Coronavirus Section 3 United States Patent. The genomic size of coronaviruses ranges from approximately delta Mately 27 to 32 kilobases, which is the longest size for any bulbul coronavirus, BuCov, known RNA virus. Thrush coronavirus, THCoV. Coronaviruses primarily infect the upper respiratory or Munia coronavirus, MuCoV, gastrointestinal tract of mammals and birds. 5 to 6.5 Porcine coronavirus, PorCoV, HKU. 15 different currently known strains of coronaviruses infect the variant replicase gene of the coronavirus of the humans. The most publicized human coronavirus, SARS, present invention may be derived from an alpha coronavirus CoV which causes severe acute respiratory syndrome such as TGV, a beta coronavirus such as MHV, or A. SARS, has a unique pathogenesis because it causes both gamma coronavirus such as IBV. Upper and lower respiratory tract infections and can also as used here in the term derived from means that the cause gastroenteritis. Middle East respiratory syndrome replicase gene comprises substantially the same nucleotide. Coronavirus, MERS-CoV, also causes a lower respiratory sequence as the wild-type replicase gene of the relevant tract infection in humans. Coronaviruses are believed to coronavirus. For example, the variant replicase gene of the cause a significant percentage of all common colds in human. 15 present invention may have up to 80%, 85%, 90%, 95%, adults. 98% or 99% identity with the wild-type replicase sequence. Coronaviruses also cause a range of diseases in livestock. The variant coronavirus replicase gene encodes a protein animals and domesticated pets, some of which can be comprising a mutation in one or more of non-structural series and are a threat to the farming industry. Economy, protein, NSP, 10, NSP, 14, NSP, 15 or NSP, 16 when compared. Cali significant coronaviruses of livestock animals include 20 to the wild-type sequence of the non-structural protein. Infectious bronchitis virus, IBV, which mainly causes respi-IBV. Retory disease in chickens and seriously affects the poultry. Avian infectious bronchitis, IB, is an acute and highly industry worldwide, porcine coronavirus, transmissible gas, contagious respiratory disease of chickens which causes gastroenteritis, TGE, and bovine coronavirus, which both result significant economic losses. The disease is characterized by diarrhea in young animals. Feline coronavirus has 225 respiratory signs including gasping, coughing, sneezing, forms. Feline enteric coronavirus is a pathogen of minor tracheal rales and nasal discharge. In young chickens, clinical significance, but spontaneous mutation of this virus. Severe respiratory distress may occur. In layers, respiratory can result in feline infectious peritonitis, FIP, a disease distress, nephritis, decrease in egg production, and loss of associated with high mortality. Internal egg quality and eggshell quality are common. There are also two types of canine coronavirus, cough, 30, CCV, 30 in broilers. Coughing and rattling are common clinical signs, rapidly spreading in all the birds of the premises. One that causes mild gastrointestinal disease and one that has morbidity is 100% in non-vaccinated flocks. Mortality have been found to cause respiratory disease. Mouse hepatitis IES depending on age, virus strain, and secondary infections. Virus, MHV, is a coronavirus that causes an epidemic but may be up to 60% in non-vaccinated flocks. Murine illness with high mortality, especially among colo. 35 The first IBV serotype to be identified was Massachusetts. Nyes of laboratory mice. But in the United States several serotypes, including archon coronaviruses are divided into 4 groups, as shown. SAS and Delaware, are currently circulating, in addition to the below, originally identified Massachusetts type. Alpha the IBV strain Baudet was derived following at least canine coronavirus, cough. 4,150 passages in chick embryos. IBV Baudet is no longer feline coronavirus, fake CoV, pathogenic for hatched chickens but rapidly kills embryos. Human coronavirus 229E, cough, 229E, H120 is a commercial live attenuated IBV Massachusetts porcine epidemic diarrhea virus, PEDV, serotype vaccine strain, attenuated by approximately 120 transmissible gastroenteritis virus, TGV. Passages in embryonated chicken eggs. H52 is another. Human coronavirus NL63, NL or New Haven, 45 Massachusetts vaccine, and represents an earlier and slightly beta more pathogenic passage virus, passage 52, during the bovine coronavirus, BCov, development of H120. Vaccines based on H120 or COM canine respiratory coronavirus, CRCOV, commonly used. In SE Asia and Micronesia, IBQX is a virulent field isolate of IBV. It is sometimes human coronavirus OC43, HCV, OC43, 50 known as Chinese QX as it was originally isolated file. Mouse hepatitis virus, MHV, lowing outbreaks of disease in the Qingdao region in China. Porcine hemagglutinating encephalomyelitis virus in the mid-1990s. Since that time the virus has crept towards HEV, Europe. From 2004, severe egg production issues have been. Rat coronavirus, ROI. Rat coronavirus is quite preva, identified with a very similar virus in parts of Western Lent and Eastern Australia where, as of March, April 55 Europe, predominantly in the Netherlands, but also reported 2008, it has been found among native and feral from Germany, France, Belgium, Denmark and in the UK. Rodent colonies. The virus isolated from the Dutch cases was identified by, no common name as of yet, COV, HKU1, the Dutch Research Institute at Deventer as a new strain that severe acute respiratory syndrome coronavirus they called D388. The Chinese connection came from FIR, SARS, CoV, 60 other tests which showed that the virus was 99% similar to the Middle East respiratory syndrome coronavirus, MERS Chinese QX viruses. A live attenuated QX, like IBV vaccine. CoV strain has now been developed. Gamma IBV is an enveloped virus that replicates in the cell infectious bronchitis virus, IBV, cytoplasm and contains a non-segmented, single-stranded turkey coronavirus, blue comb disease virus, 65 positive sense RNA genome. IBV has a 27.6 kilobits RNA. Pheasant coronavirus genome, like all coronaviruses, contains the force-struck guinea fowl coronavirus tural proteins, spike glycoprotein, S, small membrane pro.